Assalamu alaikum everyone. How are you? कैसा चल रहा है आपका? Microbiology के जो lectures पीछे हुए, any questions, any difficulties? Kindly write down in the chat box so that I have the feedback. Yes, you are listening. Can all of you hear me? बच्चों को आवाज जा रही है? So let's start with today's topic. I'm sorry for the delay. थोड़ा computer में कोई glitch थी, which has now been handled. So all of you are, I'm extremely sorry for the delay. But let's start with the topic for today. And today we are going to start with the topic of antibiotics and how these antibiotics are going to kill the bacteria. We've studied the different normal bacterial anatomy, we've studied the physiology, the bacterial growth. We've also gone through how the organisms produce disease and what are the host defense mechanisms which are going to prevent these organisms from producing a disease in an individual. But there are a number of situations where these organisms, despite all the host defenses which we have, they are going to still produce a disease in these individuals. And for that purpose, we need some treatment module, which is going to, which is going to sort of control that infection. And for that purpose, for bacteria, we have the antibiotics or the antimicrobials. If we are targeting the viruses, they would be the antiviral drugs. And if we are targeting the parasites, it will be the parasitic, antiparasitic, and for the fungi, it will be the antifungal. But today's lecture and the upcoming two lectures, they are going to concentrate on the antibiotics or the antimicrobial. So what's the difference between an antibiotic and an antimicrobial? Can anybody give me the answer? What do you understand by the terms antibiotics or the antimicrobials? And there's a difference between the two. What do you think is the difference? ये बच्चे नहीं इंटरैक्ट कर सकते, बच्चे इंटरैक्ट नहीं कर रहे हैं ना, मतलब जैसे लास्ट टाइम
बच्चों आवाज आ रही है आप सबको?
जी आ रही है।
यस आ रही है।
दोबारा बिल्कुल
आवाज आपकी
ठीक. So I was asking you about what is the difference between antibiotics and antimicrobials. What do you think is the major difference between these two?
मैम माइक्रोब्स कोई भी माइक्रो ऑर्गेनिजम्स और एंटीबायोटिक्स बैक्टीरिया के लिए.
Shabash. So basically, when we are talking about antibiotics, we are talking about things which are going to target the bacterials, bacteria, and the antimicrobials are going to target various other microbes as well. So this is a picture which is going to depict Paul Ehrlich's magic bullet concept. So this scientist had this idea in mind that there should be a magic bullet which, if introduced into the body, should go and target the particular cell which has a microbe in it. And these experiments ultimately led to the development of the drug salvaricin, which was targeting the leprosy disease. Then we came to the era of the penicillin discovery. Fleming was the individual who discovered penicillin in the 1920s. He was working on plates who had growth of Staphylococcus aureus. And what he came to realize after a few days was that there was another organism growing over here, and this was a fungus. But the interesting thing was that wherever the fungus was present, the organism was not growing. That meant that something which was being produced by this organism, this fungus, was inhibiting or preventing the growth of these bacteria. So wherever the concentration was not present, the bacteria was growing, but wherever the concentration was present, the bacterial growth was stopped. So this became the first discovery of the drug penicillin. And then it led to the discovery of other drugs. So antimicrobial agents are going to be the compounds that are going to kill or inhibit the growth of microorganisms, whereas antibiotics, they are produced by the natural metabolic process of microorganisms. And it kills or inhibits the growth of another microorganism without harming the host cells. So basically, antibiotics, when we are talking about, we are generally talking about those chemical compounds which are naturally synthesized by the other bacteria or the fungi, and they are used to destroy other bacterial structures. Antimicrobial agents, they can kill any type of organism, and they can or inhibit any type of microorganism, and they include all those drugs which are also semi-synthetic or synthetically prepared in the laboratory. This is a picture which depicts the organisms, and you can see there are a number of organisms which are producing a large group of antibiotics. And this became the first antibiotic. From there, they were made, different types of modifications were made, and we got newer and more refined antibiotics as we progressed over the passage of time. This is the timeline of the antibiotics. So this was the pre-antibiotic era, and in 1940, as I told you, it was the penicillins which were introduced, and then there were different types of antibiotics which were introduced every few years, like the chloramphenicol, aminoglycosides, glycopeptides, and this went on for a number of years. And what happened was that as the antibiotics were produced, the resistance for the bacteria against these antibiotics would develop after a short time period. And then a newer antibiotic would be required, which had a chemical structure, which was a little bit different from the previous one, so that the bacteria could be targeted. So we came down to up to 1987 or so, when we developed the different types of generations of the cephalosporins. Then we developed the carbapenems in 1993. But all of these, they were a modification of the cephalosporins, of the drugs which were produced before. And since a pretty long time, we have not had a new antibiotic. New antibiotic means that this does not belong to a class which has previously been identified or discovered. Basically, it's all the things which we have after a short time period, we were just making a sort of a change in the chemical structure of the initial antibiotic, and then we were producing a new structure and a new antibiotic. How do we classify these antibiotics? Different classification types are there. We can classify them on the basis of chemical or biosynthetic origin. We can classify them on the basis of the biological effect on the microorganisms. What is the spectrum of activity? Spectrum means which type of organisms are they going to affect, and what is the basics of mechanism of action. The most important and the most commonly utilized classification is that which is based on the mechanism of action. And most of the antibiotics we classify with keeping the mechanism of action in mind. When we are talking about the origin, we can divide them into three groups: natural, semi-synthetic, and the synthetic. So the natural would be produced by a bacteria or a fungus, and the semi-synthetic would be some modification to the natural drug, which is going to result in a newer form. For example, penicillins was the natural form, and with a sort of some treatment, they were converted into the cephalosporins. And some drugs are totally synthetic in nature. That is, they have been concocted or they have been made purely in the lab. And for example, sulfanilamide. There are a number of drugs which are purely synthetic, but majority of the drugs which we are using are natural or semi-synthetic. Based on the activity, the organisms can be divided into two groups: bacteriostatic or bactericidal. Static means to stop. Bactericidal means to kill. So all the drugs which are going to inhibit the growth of the bacteria, they are going to be put into the category of bacteriostatic. And all those which are going to kill the bacteria, they will be called the bactericidal drugs. And what happens is that the bacteriostatic drug will inhibit the growth of the bacteria, and then the natural immunity of that individual in the form of antibodies, in the form of phagocytes, will take over and will be able to control the infection by removing those bacteria from that area. So bacteriostatic drugs are not going to kill the bacteria, they are just going to inhibit their growth. They will still be, if the antibiotic concentration is reduced or removed from the area, they will again start growing. But the bacteriostatic drugs have the effect that they are going to be aided by the human immune response, and they will be able to control the infection. On the other hand, bactericidal drugs are going to kill the bacteria. These are going to affect the cell wall or the other structures of the bacteria, as a result of which the bacteria is not able to survive. It cannot perform its normal functions, so it's not going to survive. As a result of this, the bacterial cell wall is going to be destroyed, or the bacterial structure is going to be destroyed. The downside of the bactericidal drugs is that because of the release or because of the destruction of the bacterial cell wall, there will be release of toxins as well. The microbial contents of the cell will also be released. And this in turn can cause inflammatory responses and can cause damage to the tissues in the area. So when we are giving bactericidal drugs, we need to know what are the toxic effects of those drugs. Third is the spectrum of activity. Spectrum of activity, it could be a narrow spectrum or it could be a broad spectrum. Now, what do we mean by a narrow spectrum? Narrow spectrum does not mean the number. And a broad spectrum does not mean that they are going to kill more bacteria. Narrow spectrum means that they are going to be effective against a particular group of organisms. For example, penicillin, as we discussed last time, penicillin, as we are going to discuss, it has a narrow spectrum. Why? Because it is going to be effective against a very few types of bacteria. For example, the gram-positive bacteria. So, on the other hand, tetracyclines, they are going to act on the gram-positive as well as the gram-negative. So, it has a broader spectrum. So, whenever we are talking about the type of species which are susceptible to the action of the drug, we are going to call them narrow or broad. If they have a limited number, it is narrow. If they have a broad number, it is going to be a broad spectrum antibiotic. Keep in mind, it does not relate to the number of microbial species. For example, it is not relating to it kills the 100 microbes or 20 microbes or 200 microbes. And neither is it related to the potency of the drug. That is the concentration of the drug. It is not related to the spectrum of activity. So this explains the antimicrobial activity regarding the spectrum which we were talking about. For example, the penicillins, they are going to be more effective against the gram-positive bacteria, but they are going to give coverage to very few gram-negative bacteria. On the other hand, when we talk about this drug, which is isoniazid, it is going to be very effective against the mycobacteria, but it has no activity against any other organism. Similarly, we have vancomycin, which is going to be effective against gram-positive bacteria, especially the Staphylococcus and the Streptococcus. So this indicates the narrow spectrum of activity. This is also a narrow spectrum of activity. On the other hand, tetracyclines, gram-negative, gram-positive, chlamydia, different types of organisms. And this is a broad spectrum drug. Clear? Any questions? Before we proceed further, you should remember that bacteria have their own enzymes for cell wall formation, for protein synthesis, DNA replication, RNA synthesis, and also for the synthesis of essential metabolites. So when we are taking into consideration the development of an antibiotic, or when we are doing experiments on the antibiotic, we have all these targets which can be destroyed to destroy that particular bacteria. Just a recap of what we did before. Peptidoglycan molecule, as you know, it is made up of these layers of NAM and NAG, and they are interconnected with each other. And then these layers are also interconnected with each other through the peptide linkages. And in case of gram-positive bacteria, we have both. We have a large number of these layers, and these small spaces are also present inside them, which will allow the antibiotics to pass through. On the other hand, when we talk about the gram-negative bacteria, they have a very thin layer of peptidoglycan. So therefore, those antibiotics which are targeting the peptidoglycan layer will not have a very good effect on the gram-negative bacteria. On the other hand, gram-negative bacteria has an outer membrane. And as you must recall, it is made up of lipopolysaccharides, phospholipids, and proteins, and it is a barrier to the diffusion of molecules, including the antibiotics. Some antibiotics may diffuse through, some may pass through the porins, but therefore, because it has this barrier, the antibiotics are less effective against the gram-negative bacteria until and unless they are targeting the outer membrane. Remember, there is no molecule similar to peptidoglycan in humans. So when we make a drug that targets the cell wall synthesis, it becomes very selective. And it becomes selective in its toxicity against the bacteria. And it generally does not produce harm to the human tissues by itself. The harm can be produced by the process of phagocytosis or the immune mechanism which is taking place, but the drug itself does not have any effect on the cells of the human individual. If you remember your anatomy, bacterial anatomy, then it is going to be very easy for you to remember the targets of the antimicrobes. There are 5 different targets, or 6 different, basically there are 5 different targets. And if we go from the outside to the inner side, it will be very easy for you to remember. Number 1 are the inhibitors of cell wall synthesis. So there are certain drugs which are going to prevent the synthesis of the cell wall, which is made up of peptidoglycan layer or the outer membrane and the lipopolysaccharide. And in this category, majority of the drugs are going to be effective against the gram-positive bacteria because these drugs, they are targeting the peptidoglycan layer. Then we have the cell membrane interior to the cell wall. There are certain drugs which are going to target the membrane, and these drugs are going to cause disruption of the membrane function, and they are then going to cause a defect in the cell membrane and the cell wall, and they are going to cause the bacteria not to survive in that environment. Then we have the inhibition of translation and transcription. These are going to affect the RNA synthesis. That is basically the process of transcription and translation, and they are going to be involved in the in inhibiting the transcription and the translation processes. And then we have the inhibition of DNA replication, and finally the metabolic processes which are going on in the bacteria. We have the inhibitors of metabolism. So different drugs, different categories, need to remember that there are 5 basic categories: inhibitors of cell wall synthesis, disruption of these drugs which are going to cause disruption of the cell membrane function. Then they are inhibitors of translation and transcription or RNA synthesis. Then they are inhibitors of DNA replication. And finally, they are inhibitors of metabolism of the bacterial cell. Now, first of all, let's talk about the drugs which are affecting the cell wall. We have 2 groups: beta-lactam antibiotics, and we have certain other antibiotics which are chemically different from the beta-lactam antibiotics. Amongst the beta-lactams, you have the penicillin group, the cephalosporins, which are further subdivided into 5 generations. We have the carbapenems and the monobactams. In the other antibiotic category, we have the bactericin, vancomycin, daptomycin, and then there came a time when the bacteria started developing an enzyme which was known as the beta-lactamase enzyme. And this beta-lactamase enzyme was destroying the beta-lactam antibiotics, and the bacteria became resistant to these antibiotics. For that purpose, we needed a drug which could control the beta-lactamase enzyme. And there we were with the production of the beta-lactamase inhibitors. So if a bacteria is producing the beta-lactamase enzyme, it is not going to be controlled by any of these drugs. For that, we need to give the beta-lactamase inhibitors, which are going to destroy the lactamase enzyme, and then the antibiotic would be able to have an effect on the cell wall. Clear? Any questions?
No, ma'am.
Okay, then we come to the group penicillin. First, we'll discuss a little bit about this. There are more than 50 compounds, and the common thing is this 4-membered ring, and this is known as the beta-lactam ring. And this beta-lactam ring is going to form the basis of the natural as well as the semi-synthetic penicillins. So amongst the natural penicillins, we have penicillin G and penicillin V, whereas in the semi-synthetic penicillins, we have the extended spectrum antibiotics like the ampicillin. Then we have the antibiotics which are going to be resistant to penicillinase enzyme produced by the bacteria like the oxacillin. Because the cell wall of the bacteria differs from that of the protozoa, fungi, viruses, and well as Mycobacterium tuberculosis or other mycobacteria, these penicillins are not going to be effective against all these 4 groups. You need to remember that the Mycobacterium, protozoa, fungi, and viruses, they are not going to be treatable with the penicillins. Penicillin antibiotics are divided into 6 main groups: natural penicillins, carboxypenicillins, beta-lactamase resistant penicillins, uridopenicillins, aminopenicillins, and the penicillin inhibitor combinations or the beta-lactamase inhibitor combinations. You need to remember one, or you need to remember at least 2 amongst each of these groups. So before we go further on the mechanism of action of these penicillins, let's talk about the penicillin binding proteins. Penicillin binding proteins are a normal constituent of many bacteria. They are a sort of an enzyme, and they belong to the group of transpeptidases. Basically, penicillin binding proteins, as we discussed when we were talking about the cell wall synthesis, they are involved in the final stages of the synthesis of peptidoglycan layer. And these penicillin binding proteins, they are going to be essential for the growth and reproduction of the bacteria, as well as for the maintaining the cell structure, because without these penicillin binding proteins, the peptidoglycan layer will not be formed, and therefore the growth, division, and the cell structure would be destroyed. These are the this is the penicillin binding protein. There are different types. You just need to remember that this is the peptidoglycan, the penicillin binding protein. And as we discussed last time, the sugars as well as the pentapeptide bridges, they were synthesized in the cytoplasm with the help of a carrier protein and a lipid molecule. They were crossing the cytoplasmic membrane, and then this is going to cause a nick in the peptidoglycan layer, and this new would become inserted into it. So in this way, this synthesis keeps on going, and the layers which are going to be older, they are going to be discarded, and newer layers are going to be added to the cell wall. Now, what does penicillin do? It inhibits the formation of peptidoglycan cross-links in the bacterial cell wall, especially in gram-positive organisms, because that is the thickest peptidoglycan layer which a bacteria has. So first, it is going to bind to the penicillin binding proteins. Then it is going to interfere with the transpeptidation reaction, and this is going to over here you have, this is the transpeptidase domain, and what it is going to do, it is going to get attached to this. And when it gets attached to this, it is going to interfere with the transpeptidation. And when it interferes with that, the peptidoglycan synthesis is going to stop. So penicillins are bactericidal, and they are going to act only on the growing cells. They are not going to be toxic to the human cells or animal cells because we do not possess a cell wall. Now, beta-lactam antibiotics bind to the penicillin binding proteins because the penicillin binding proteins and the beta-lactam antibiotics, they are similar in structure to the peptidoglycan. So when we introduce a beta-lactam antibiotic in the environment where the bacteria is present, instead of binding to the penicillin binding proteins, it is going to block the penicillin binding proteins. Rather, it is going to bind to the penicillin binding proteins, and it is going to block those proteins. And the structure which is then going to go and get attached to the peptidoglycan layer, that would be blocked. So again, the transpeptidation process would be stopped. And basically, when the beta-lactam antibiotic binds to the PBPs, the beta-lactam ring opens, and once the ring is open, then the penicillin becomes inactive. This is an irreversible reaction, and it also inactivates the enzyme. Once the PBPs are becoming inactive, they cannot synthesize the peptidoglycan layer. This is going to result in the defects in the cell wall structure. There will be irregularities in the cell shape, and there will be the formation of spheroplasts. Spheroplasts is basically when the cell wall is going to be destroyed. There are going to be openings in the cell wall. The fluid from the outer environment is going to go into the inner environment. If the osmotic pressure does not balance, then the cell is going to burst. And therefore, we have an antibiotic which is causing the bursting of the cell. Clear? Is this concept clear?
Yes, ma'am.
How the penicillins act?
Yes, ma'am.
Okay. Now, there are three ways in which these penicillins are acting, two ways rather. One we've already discussed, and the other one is the inactivation of the autolysins which are normally being produced by the bacteria. So normally the bacteria have autolysins within them, and they are inhibitors also present within the bacteria which prevents the autolysins from being released and destroying the bacteria. But penicillins also inactivate that inhibitor, as a result of which autolysins start to be released from the bacterial cell. And apart from the activity of the penicillin binding proteins being blocked, the autolysins which are being produced by the cell, they are going to kill that, not only that bacteria, but also the other bacteria which are going to be present in the environment. So even if one or two bacteria get bound by the beta-lactam antibiotic, the others can still be destroyed with the release of the autolysins. They have little activity against the gram-negative rods, and they have more activity because they can bind the peptidoglycan layer. They can bind to the penicillin binding proteins. In the gram-negative rods, they cannot penetrate the outer membrane. Now, we were talking about the enzymes which are being produced by the bacteria. This is known as penicillinase or beta-lactamase. When this enzyme is being produced by the bacteria, it is going to act on the penicillin, and this beta-lactam ring, that would be destroyed. So the active penicillin becomes inactive. Now, this inactive penicillin cannot bind to the penicillin binding proteins. It cannot induce the reduce the or remove the inhibition of the autolysins. So consequently, the penicillinase binding proteins, the penicillinase producing proteins, they are going to be resistant to penicillin. So this is one mechanism which the bacteria have developed for resistance to develop against penicillin. And this developed in the early 1950s. Therefore, we went back to the lab and we went back to the production of newer antibiotics. Just to explain the function of the beta-lactamase on the penicillin binding proteins. Over here, you see a normal process happening. This is the gram-negative, this is the gram-positive. In the gram-negative, through the pores, these are the pores, and normally the peptidoglycan subunits are being synthesized and they are causing the synthesis of the peptidoglycan. Similarly, over here, if you introduce the beta-lactam drug, this beta-lactam drug is going to go bind to the penicillin binding protein, and the penicillin binding protein gets locked, so these subunits cannot get attached to it, and therefore the synthesis of this peptidoglycan layer is stopped. Also, it is going to remove the inhibitor of the autolytic enzymes, and these enzymes are going to cause further damage to the cells. This was about the gram-negative bacteria. When we talk about the gram-positive bacteria, we have this peptidoglycan layer. We can have a cell wall or the cell envelope, and we have or a capsule, and then we have the beta-lactamase enzyme being produced. When the beta-lactamase then is produced, this is going to block the beta-lactam drug. Now, this beta-lactam drug cannot go and bind to the penicillin binding proteins. Consequently, the organism becomes resistant to the beta-lactamase enzyme. The same thing happens in gram-positive bacteria. So, without the lactamase production, both in gram-positive and gram-negative bacteria, we are able to destroy the cell wall. But with the presence of beta-lactamase enzyme, which acts as a blocking agent for the beta-lactam drug, neither the gram-negative nor the gram-positive bacteria are going to be destroyed, and both of them will develop resistance against the penicillin. Any questions?
No, ma'am.
Understood? So, when this happened, we needed an antibiotic which could inhibit this beta-lactamase enzyme. And for this, we needed certain chemicals which could destroy the beta-lactamase enzyme, and these are lavulonic acid, tazobactam, and selbactam. And these would bind with the beta-lactamase and prevent the enzyme from breaking down the penicillin. So as we were discussing in the last over here, the beta-lactamase is binding with this penicillin, it is binding with the beta-lactam drug. But if we are going to produce a drug which cannot bind over here, then this penicillin will remain free to act on the cell wall. So these combinations were prepared which were having a penicillin group, and then they were having these chemicals, and they were marketed for different names, and these were penicillin beta-lactamase inhibitor combination drugs, and they were effective against all those infections in which penicillin groups were going to be resistant. Just to recap that the beta-lactam antibiotics have a beta-lactam ring, and there is a 5-membered ring attached to it in case of penicillins, and a 6-membered ring is going to be attached in case of the cephalosporins, which are the second group of drugs which are inhibitors of cell wall synthesis. So these were synthesized naturally as well as semi-synthetically. How do the inhibitors of cell wall synthesis are going to be proved? These were first introduced in 1965 and the 5th generation in 2013. So as the first generation was produced, it had activity against the gram-positive, but very less activity against the gram-negative. So that propelled the pharmaceutical industry to produce a second generation, which had an expanded spectrum of activity, which had a better cell penetration, and which also had a resistance to the beta-lactamase enzyme. But in this process, the potency against the gram-positive bacteria, or the effectiveness against the gram-positive bacteria, became less, although they were more effective against the gram-negative. So then we started using the first generation for gram-positive infections and second generation for the gram-negative infections. Again, resistance developed, and the third generation came into the picture. It again had a better activity against the gram-negative, better cell penetration, and a better target binding. Then we came to the 4th generation, more effective against the beta-lactamase producers, and finally the 5th generation, which was approved for treatment of critical infections, because these till the 4th generation, the resistance was being seen in especially hospital-acquired infection organisms. Then the other third group of the penicillin is or the beta-lactam antibiotics are the carbapenems. Carbapenems include imipenem, ertapenem, meropenem, and the doripenem. And they have different spectrum of activity against different organisms. Imipenem is more active against gram-positive bacteria. Ertapenem and meropenem, they are more effective against the gram-negative bacteria. And for doripenem, we are basically looking for an antibiotic which is going to give us CNS coverage, whatever the organism, because it has a better penetration in the nervous system. And also it is going to be effective against a gram-negative organism, Pseudomonas, which is resistant to most of the antibiotics. Certain inhibitors of cell wall synthesis are going to be different from the drugs which we have just discussed, because the cell wall of that bacteria is different from the cell wall of the gram-positive or the gram-negative organisms. And these antibiotics, they are going to be effective against the mycobacteria. For example, Mycobacterium tuberculosis or leprosy, they have a cell wall which is going to be entirely different from the cell wall of the gram-negative or gram-positive. You can see the structure. We study it in detail when we are studying the mycobacteria. And this has a lot of glycolic acid in it. And the drugs which are going to target the mycobacterium should be those which are going to interfere with the glycolic acid synthesis or the incorporation of glycolic acid into the cell wall. And these drugs were prepared separately, and they are just going to target the mycobacterium like the isoniazid and ethambutol. So we have a special class of drugs which are going to be effective against the mycobacterium. Cycloserine is a structural analog of D-alanine, and it is going to inhibit the synthesis of the cell wall dipeptide D-alanine D-alanine. And it is a second line of drug in the treatment of tuberculosis. But basically, it's the first line of drug. If they are going to be less effective, then we introduce cycloserine. Otherwise, we don't. And we'll just go back to this diagram, just to show you, give you a recap. What is happening with the different types of antibiotics. Over here, you have the cytoplasm. In the cytoplasm, we have the synthesis of cell wall precursors, NAM, NAG, D-alanine. So cycloserine is going to inhibit the reaction, which is going to involve the incorporation of alanine into the cell wall precursors. Then, when we move on to the cytoplasmic membrane, that is the place where the new cell wall subunits are going to be attached, and they are going to be we have two types of antibiotics which are working. Number one is the bacitracin, and this bacitracin is going to inhibit the dephosphorylation of this phospholipid carrier. So when this is not going to be available, it is not going to get attached to the unit, and it is not going to carry it across the cell membrane. So basically, there is a deficiency of the lipid carrier because bacitracin is preventing the dephosphorylation or the removal of the phosphorus group from this lipid carrier molecule. So therefore, the synthesis is going to be blocked. And then we have the glycopeptides, which are going to bind to the terminal D-alanine residues, and therefore they do not become incorporated into the subunit of the peptidoglycan layer, and therefore this is not going to go into the cell wall for attachment. And in the cell wall, we've discussed the beta-lactams, which are going to bind to the penicillin binding proteins and prevent the growing of the peptidoglycan layer. So these are the four groups of drugs which are going to affect from the cytoplasm right up to the cell wall and inhibit the cytoplasmic and inhibit the cell wall synthesis. A few words about bacitracin. It's a bactericidal drug. It is a polypeptide antibiotic. It's a natural drug which is produced by a bacteria known as Bacillus subtilis. It is effective against the gram-positive bacteria and 2 gram-negative bacteria, Neisseria and Haemophilus influenzae. And as we've discussed, it prevents the dephosphorylation of the phospholipid that carries the peptidoglycan unit across the cell membrane. Therefore, the lipid carrier is not regenerated, and therefore we have the cell wall synthesis inhibition. It is not absorbed orally, and when given IV or parenterally in the form of an injection, it has a very high toxicity for the kidney. Therefore, bacitracin is utilized only as a topical application. Topical means it is just applied on the outside onto the skin, intact skin, not on the broken skin. So it is going to be given in different types of infections like the eye infections or skin ulcers. And it's not going to be given orally or parenterally because it will cause extensive damage to the kidneys. Then we come to the second group of antibiotics which cause disruption of the microbial cell membrane. And these are known as polypeptide antibiotics, which are targeting the bacterial membrane as their mode of action. They are low molecular weight polypeptides. They have a lot of toxicity because of the bactericidal effects, and they are produced by various types of bacteria. Most important, which are going to act on the cytoplasmic membrane, are the polymyxin B, which is produced by, again, a bacillus species. And then we have a colistin, which is polymyxin E. Again, it's a bacillus species. Here we see how these antibiotics are going to cause disruption of the cell membrane or the cytoplasmic membrane. These polypeptides, they are lipophilic, that is, they have an affinity for the lipid portion of the outer membrane, and they have detergent-like properties. Once they get attached, they are going to cause the disintegration of the outer membrane. When the outer membrane gets disintegrated, more the polypeptide molecules can go into the space between the inner and the outer membrane, and then once they reach the inner membrane, they are going to cause the same action, cause disruption, and then they can go into the cytoplasm and cause damage to the interior structures. Now, we've discussed the mechanism of action just to show you that their polymyxins have three number polymyxin B, which is effective against the gram-negative bacteria, but very few, and but it is effective against the multi-drug resistant strains, and it has a topical preparation. Polymyxin E is colistin, and this is utilized for the multi-drug resistant strains, especially in individuals who are undergoing the severe bacterial infections, who are undergoing surgery, and they require a cover which is going to cover a large number of organisms which may be resistant to various antibiotics. And finally, we have a lipopeptide, which is the daptomycin, and this is a narrow spectrum against the gram-positive bacteria. Colistin, it's again a polymyxin and acts on the bacterial cell membrane. The only thing which I added over here, this slide over here, was to tell you about the potent endotoxin activity. So colistin has, just like the lipopolysaccharide of the gram-negative bacteria, it has the endotoxic activity, and this is going to cause the damage not only to the bacterial cell but also to the tissues in the humans. And it is a last resort treatment for multi-drug resistant gram-negative infections. What you need to remember is that the membrane targeting mechanism is not a selective toxicity. When you are targeting the cytoplasmic membrane, you are also targeting the cytoplasmic membrane of the human cells, and they are going to damage the cells in the human system, maybe the kidney, the nervous system, when they are going to be administered systemically. Because of these serious side effects, they are also, and they are poor absorption from the gut, they need to be given intravenously, and because of the high toxicity, they are going to be restricted to use as a topical antibiotic, especially polymyxin B, which has the highest toxicity for the kidneys. Any questions? Okay, we'll start with the inhibitors of protein synthesis next time, tomorrow that is, and we'll continue from here. If you have any questions, I would like to address them at this point. No questions? That means you've understood everything, which means that I should ask you. Yes, Rita. Any questions, Rita?
No, ma'am. समझ आ गई है.
चलो, आपको तो बेटा समझ आ गया. Anybody else?
No, ma'am.
No, ma'am.
Okay. please go back and do give it a reading, even if you don't remember anything, but do give it a reading. It will clear your concepts, and if you have any questions, you can ask me tomorrow. Thank you so
मैम
much. जी
एलएमएस
बेटा।
के ऊपर स्लाइड्स जो हैं आप पोस्ट करते हैं वो हमें ज़्यादा आसानी हो जाती है कि क्योंकि इसके अंदर समराइज मटेरियल होता है, जब बुक से स्टडी करते हैं तो दोबारा इसको गो थ्रू करने में ज़्यादा आसानी होती है।
बेटा आपकी बात सही है, आई विल पुट देम अप बट नॉट एट दिस स्टेज। अभी आप किताब अपनी पढ़ो, because what I have come to learn over the years is that when you are given these slides, then your books get pushed to one side और आपकी सारी की सारी स्टडी जो है ना वो इन्हीं स्लाइड्स तक रह जाती है, which are insufficient. ठीक है, they are the bullet points which I talked to you about. So I will definitely give them to you, but first you go through these chapters and come up with questions, and once you are through with them, then I will give them to you as a backup for just revision. Sounds good?
ओके मैम, लेकिन मैं बस ये कह रहा था कि ये होता है कि हम दो चीज़ें जब कंपेयर करके, ऑब्वियसली हमारा बेसिक मेथड ऑफ स्टडिंग जो है वो तो स्टैंडर्ड टेक्स्ट बुक होनी चाहिए, बट दोनों चीज़ों को कंपेयर करके मतलब हम ज़्यादा पॉइंट्स बेहतर तौर पे बना सकते हैं। जैसे यहाँ पे हेडिंग लगी होती है और यहाँ पे समराइज टॉपिक्स जो मेन पॉइंट होता है वो यहाँ पे हाईलाइट हुआ होता है, तो बुक्स के अंदर वो हमारे लिए कंपेयर करके स्टडी करना ज़्यादा आसान होता है।
ठीक है बेटा, ठीक है। I'll see कि मैं इसका आपको जो है ना put them up. I'll just give me some time. ठीक है, I'll put it up.
थैंक यू मैम। थैंक यू मैम।
Any other question? Thank you, beta. Thank you. Take care.